Good afternoon, ladies and gentlemen. Now let's welcome the chair of this session, Academician of Academia Sinica, Dr. Andrew Wang.
Okay, thank you. Let's start the afternoon session 2 on SARS coronavirus 2. And we have 4 speakers today. Now, Dr. Shih will give the lecture in person, but the other 3 will be virtual. So let me introduce Professor Shin-Ru. She is from the Department of Medical Biotechnology of Chang University, but she's also a professor there, the director of Research Center for Emerging Viral Infections, Chang University. And she is a leader in this field. In fact, my understanding is that her lab is one of the reference labs on viral infections in Taiwan. So she plays a very important role in the current COVID-19 pandemics. So let's welcome Professor Shih. She will give a talk on genetic variation of SARS-CoV-2, impacts on diagnosis, antiviral, and vaccine development. Let's welcome her.
My talk here will include 3 parts. The first is about genetic variation of this virus and its impacts on laboratory diagnosis. And I will show you the genomic surveillance of this virus in Taiwan and a comparison with global trends. And I will also talk about diagnosis specificity and sensitivity. And we figure out not only sequence, but viral genome integrity, also important for virus isolation. And the second part, I was talking about the impacts of viral genetic evolution on antibody and drug vaccine development. And of course, this part will focus on the variation of the surface protein, spike protein. And the third part is about the mutation on drug targets for antivirus. And I'd like to focus on RD-RP, RNA-dependent RNA polymerase, and protease. Okay, we heard about the surveillance, and we know Taiwan is lucky. And until now, we only have 5 more hundred cases, and most of them are imported cases. So we only have a limited number of sequences. It's around 60 sequences here. But I believe because we are all imported cases and only few local cases, so it's kind of our sequence reflect the sequence around the world. And here I'd like to show you a study. Even though our sequence number is limited, we also found some diversity in Taiwanese isolates. And first diversity is that we found 382 nucleotide deletion in open reading frame 8. And this deletion not only was found in Taiwan, but also in Singapore group. And we, on our hands, in cell culture level, we did not find any effect on viral RNA replication, but in Singapore group, they did the animal study and they found without ORF8, the virus is kind of attenuated. And through our sequence analysis, especially for our local strains, we found a new clade of several strains, Taiwanese strains, harboring ORF1AB, V378I mutation. And this group kind of similar to some strains from patients having a travel history of Iran and Turkey. So we believe the genomic surveillance is very important, especially in Taiwan. All of them are imported cases, and we need to set up such kind of sequence frame. And just in case we have some local strains, we can trace the origin of the infection. Okay. And for this study, for the ORF8 deletion, when we analyze the first 2 isolates in our hospital by next generation sequencing, we found around the end of the genome, there is some difference. We further use primers and to sequence the PCR product by single sequence, and we defined there is some deletion here. And when we use the primer, as I said again, we kind of validate there is a deletion, and it's a ORF8 deletion. And this is the phylogenetic tree analysis of our isolates. And basically, we can divide it into 4 clades. And somebody have further divided. But our definition is that when we found one signature can retain at the clade, we kind of define there's a new clade. And here for the gray part, for the I think the origin circle indicated the Taiwanese isolates, and we have some local isolate can be divided into a new clade, the C clade here. And I think probably this figure can kind of explain our isolates more clearly. In general, we got imported cases. So if the isolates from the patient that came from European countries, their sequence is kind of similar, close to the isolates in European countries. And similar the case for China, Asian country, and the United States. And here we have a hospital clustering in our hospital. And we found it's kind of quite different from the others, different from the patients came from United States or European countries. They cluster together and cluster with some patients having history to Middle East countries. So we would like to know, does the sequence variation affect the sensitivity and the specificity for laboratory diagnosis for COVID-19? And so here we analyze the primer region of our diagnosis method. The primers we use suggested by Taiwan CDC, also quite similar to WHO and also other countries. And they are located on the RDRP gene here at this region, and also the E gene here. And some laboratory also use N gene and other RDRP gene as diagnostics methods. And here we use entropy to indicate the sequence diversity. And if the entropy is lower, near 0, indicating the sequence is kind of very conserved and no variation, and indicating the primer we use still can detect the recent isolates for COVID-19. So we found in RDRP regions, also in E primer regions, there are not too much difference. So the primer we use currently still work. And there are some variation at this position, but not is in the middle of the primer. So no effect on the laboratory diagnosis. So what's the effect for laboratory diagnosis in terms of sensitivity and specificity? So we did not find any sequence variation got effect on laboratory diagnosis, but we found the genome integrity is important for diagnosis. So here is our finding. Through January to March, we got 50 patients, and we collect their specimens either from throat swab, nasopharyngeal swab, or sputums. And some of the specimens have underwent frozen and thaw procedures. And it has been known such kind of procedure may break viral RNA. And so we did a culture. And actually, we want to know a question which has been mentioned by Dr. Wang the previous talk. We don't know whether the live virus exists in the specimen or not. And it's kind of very important because it will decide whether the patients can be discharged and also can decide the quarantine time. And I'd like to thank those people in our hospital do the laboratory diagnosis. It's kind of frontline people. And also thank Dr. Li and Dr. Huang for this study. They are major contributors. And as expected, this the y-axis indicating the log number of RNA copies. And for the x-axis is the we use RDRP gene or E gene or N gene. And for figure A is culturable specimen, and figure B is non-culturable specimen. So in general, we found the culturable specimen the RNA copies existing in the specimen kind of higher than a non-culturable, but there are some overlapping here. And the primers we use, similar to Taiwan CDC and also WHO, is RDRP gene and also E gene. And we also try another gene, N gene here. E and N gene, both of them are subgenomic RNA. In general, they are more abundant than the genomic RNA, such as RDRP gene. And for the culturable specimen, they are associated with higher correlation of copy number. And we also found that genome integrity is very important to determine whether the virus is live or not in the specimens. Here again, the y-axis is the copy number of RDRP gene, also called NSP12. And for the x-axis is the subgenomic RNA, either E or N. And the red line indicating those specimens are culturable, indicating we can go to P3 lab and obtain the live virus. The blue line is non-culturable. So if we can see the red line, the correlation of genomic RNA and subgenomic RNA correlation is better for the culturable specimen, better than the non-culturable specimen. And similar finding to the subgenomic RNA, RDRP2, the genomic RNA, RDRP2 subgenomic RNA N. And for this figure, I think particular obvious because those specimens were from frozen thaw specimens. So some of the viral RNA could be broken down. So for the culturable one, we still can see very good correlation near 1.0. But for the non-culturable RNA, for the correlation between genomic RNA and subgenomic RNA is quite low, indicating the RNA may be broken down. So the genome integrity of specimen should be considered whether the patient contains the live virus or not. Okay, so here for the summary for this study, we found the lowest copy number required for virus isolation. And it's kind of a little bit different from different genes, especially for genomic RNA and subgenomic RNA. And we did a culture for each specimen, and we found culturable specimens are characterized by high copy number of a linear correlation observed between copy numbers of epicon. Targeting structural and non-structural is the genomic RNA and non-genomic RNA. And in addition to copy number, the integrity of viral genome, because from the frozen thaw specimen, we did observe when the RNA, kind of broken RNA, we can not culture the specimen successfully. So our viral genome integrity should be considered when evaluating the infectivity of the specimen. Not only infectivity, we also found for RT-PCR, if the RNA is kind of broken, and then the sensitivity and the specificity for real-time RT-PCR also become low. Okay, now I'd like to move to the second part. It's about the impacts of viral genetic evolution on antibody, drug, and vaccine development, and focus on surface protein, spike protein. It determines infectivity, antigenicity, and stability. So at that time, I think in the early of February, our physician doctor, Dr. Huang, who isolated the B cell from a patient and did the cloning, transfection, expression, and purification, we obtained a set of monoclonal antibody. And we tried the binding assay, and for one of the antibody, we actually tried many antibodies, and this is one of the example, EY6A. And this is another monoclonal antibody obtained from SARS patients 17 years ago. And here, the black line indicating the binding affinity for SARS-CoV-2, either whole spike. The red line is S1. And for SARS-CoV-1, 17 years ago, is a blue line, MERS is green, OC43, another coronavirus, is in, orange. So for the antibody isolated from SARS-CoV-2 patient, we found the antibody got high affinity with SARS-CoV-2 spike protein, and not that good for SARS, but still have binding affinity, and none for MERS and OC43. And for this SARS-CoV-1 antibody, got the greatest binding affinity for SARS, and also have some binding affinity with SARS-CoV-2, also known for MERS and OC43. This is another assay, immunofluorescent assay, to prove this finding. And we did the neutralization assay, also plaque reduction assay at P3 laboratory. And when we add this antibody against SARS-CoV-2, and we did the real-time RT-PCR, we can see the CT value shifted lower from higher, indicating the RNA copies are reduced upon the treatment of this antibody. And this is for plaque reduction assay. When we increase the amount of the antibody, we can see the inhibitory effect kind of increase almost to 100%. So the antibody is a neutralizing antibody. And in comparison with the SARS antibody, the SARS-CoV-2 antibody got a better inhibitory effect for SARS-CoV-2 virus. And this has been done by Dr. Huang at P3 lab. And in the collaboration with the structural biologist in Oxford University, and we got the structure of the complex is EY6A, one of the antibody against SARS-CoV-2, and this is receptor binding site for the virus. And indeed, this antibody can block the receptor binding site. So this is why in vitro and cell culture assay, we did observe the neutralization ability. And the binding position not only receptor binding site, but also covers several residues outside of the receptor binding site. This is our finding. So we would like to ask a question: are these human monoclonal antibodies still effective to other strains of SARS-CoV-2? And on our hands, we tried several isolates. Later on, not only the isolates, the patients came back from Asia, but also from European countries and United States, and all of them can be inhibited by this monoclonal antibody. And when we did the sequence diversity analysis, I think I kind of can support our finding. We download near 100,000 sequences from GSAT and trim gaps and ambiguous nucleotide positions. Again, we calculate entropy for the to present the diversity. And here is the whole genome. And basically, we can see on the spike region is kind of more diverse, but also not that much. Expect the position 6 or 14 is from D to G. I think is a famous mutation. We can discuss this later on. And the other 4 regions got higher entropy, indicating more diversity, sequence diversity in their regions. 2 are in ORF1AB, actually 3. Okay, and this is within RDRP gene. And 4 is the D614G, as I mentioned, and another one is at ORF3A. ORF3A has been known probably contributes to the virus-induced apoptosis and may relate to some immune modulation. So we look at in detail for the spike protein. The 3 major positions got some diversity, but actually the entropy for the other 2 are kind of below 0.2. Only D614G, I think the entropy is higher. And this is the position we can see here. It's not within the receptor binding site. It's outside the receptor binding site and near the S1 and S2 junction. And D16-4G, I think many groups have done researches for the impact of this variation already. And as you can see here, the cover it all published in Cell. They found the global transition at this position, D to G, I think began around March and now become the dominant one. So the consistent increase of G16 at region I think may indicate the fitness of advantage. So this is why the D to G mutation of the virus can return and become the dominant strain. And in patients, they found if the patient got G infection, the mutated one, they have a lower level of RT-PCR CT value, indicating the RNA number is higher, the viral load is higher in patients. And also they used a pseudovirus, which only one spot, this spot, 16-14G in spike protein are different, D to G difference. And they found that G, the mutated one, the variance, grows to a higher titer as the pseudovirus variants. And since many people have done this research, and here on our hands, we would like to ask the question, how about the stability? So we compare these two strands. One is the early strain, which contained D16 at this position, 14. And another strain is G. But actually, when we look at in detail for this, the sequence difference in these two strains, not only D16G got difference here, but also have several difference at 5' UTR or ORF1AB or N genes or 3' UTR. And we compare the stability of these two viruses. And when the virus stuck at -80, is the general temperature for virus storage. We did not see too much difference. But when we use different titer of these two viruses and store at -20 over 4 degrees, which is kind of common temperature for food storage. So here in at low titer of the virus, we can see the old strain, the CDC4, the early strain, the stability is kind of very poor. The viral titer decline quickly after so many days. Okay, and even though we increase the titer, we can see the viral titer increase. We can see the stability is better. But again, for example, such as at 4 degrees for the CDC 179. Which harbor the G at spike protein 614, the stability is kind of very poor. And when we detect the viral RNA expression, we observe the similar results. But it's kind of surprise to us because we think probably viral RNA will keep this quite similar because a spike protein won't affect RNA stability. But since, as I mentioned, these two strains not only harbor this difference in spike protein, but also have some difference in other genes. For example, the nuclear capsid protein. So probably the mutation at those positions may affect the RNA stability. So this is why when we store at different temperatures, we see the decrease of the RNA amount for the CDC 179, which harbor G at spike protein 614 position. Okay, and finally, I'd like to spend only a few slides to talk about the drug targets. And as we know, such as remdesivir, it targets to RDRP gene. And some I think, developing drugs are targets to protease. So how about the diversity of these two genes? Again, here we use entropy to indicating the sequence diversity as in these two regions. And we found several peaks which have higher entropy, indicating the diversity is higher. And when we look at the remdesivir target gene, RDRP, we can see these two genes. But so far, nobody to test the accessibility of remdesivir for this kind of new strains. I think probably it is worth to do whether such kind of mutation will affect remdesivir accessibility. And another one is protease. It's a 3C-like protease, and I think many developing drugs targeting to these genes. And we only found very low mutation rate at this region, and it is not a catalytic size. So in general, even though there are some diversity in this part, it won't affect the accessibility of the drug to a 3C protease. So here I would like to give a summary of my presentation. SARS-CoV-2 genomic surveillance in Taiwan reveals novel ORF8 deletion mutation. And we also found a nucleate associated with infection in Middle East countries. So far in GSET, not many sequences available from Middle East countries. And the primer design in January is still working for laboratory diagnosis, real-time RT-PCR, no problem, because of low sequence diversity in the primer design regions. And there is only one little bit diversity, but it's within in the middle of the primer, so I don't think it will affect the real-time RT-PCR assay. And the monoclonal antibody we isolated from a recovered patients infected in February and still can neutralize it. Recent SARS-CoV-2 until not recently we got several isolates from US or European countries, and it still can work. But there is a mutation in the spike region. It's D614G, and it has been proven by many groups to affect viral infectivity, and here we also found it may affect virus stability. And only low sequence variation are found in two drug targets so far, the RDRP gene and protease. So I will stop here. Finally, I'd like to acknowledge the Chang research team and also collaborators from Taiwan CDC, NHRI, Academia, Sinica, Oxford University, and Rutgers University. Thank you.
Thank you, Professor Shih. In view of the time, I think we'll allow one question. Any question from the audience? Maybe I'll ask one question. Recently, I think many people tested negative in Taiwan, but when they go to other countries, they became positive. Do you have a reason for that?
I think first, we did not check that patient, exactly the same patients, so it's hard to say. And another thing, as I know, we set CT value such as 35 as positive or negative. And personally, I think probably we can set 40. It will be safer. And but we did not know what kind of assay they use, such as Philippines, right, or other countries. Yeah.
Okay, thank you, Professor Shih. Okay, the next one is, I think it's a virtual one, right? So from Stephen Burley, he is a prominent structural biologist. He and I actually worked at MIT together a long time ago. I won't say how long, but anyway, currently he is a university professor and Henry Rutgers Chair in the Rutgers State University of New Jersey. And he actually now plays a very important role. He's director of the PDB data bank. And so I invited him to give us a view on this structural biology of big data in the current pandemic. So what can we learn from all those structural informations? So let's hear what he has to say. Okay.
Good afternoon. I'm honored to participate in this joint Academia Sinica CODATA meeting. Let me begin by thanking the organizers for their kind invitation. Both the subject matter and the worsening pandemic in the US make me wish that I was attending in person. Before proceeding with my talk, I want to thank academician Andrew Wong for his kind introduction. Lao Wong taught me small molecule crystallography when I was a visiting graduate student at MIT. I also need to acknowledge the successes that the people of Taiwan and their government have had managing the COVID-19 pandemic. Every country should have a citizenry that respects science and a vice president with a PhD in epidemiology from Johns Hopkins University. My subject today is the Protein Data Bank in the time of the coronavirus. I will describe how we've used the contents of the PDB archive to analyze the evolution of the SARS-CoV-2 proteome in 3 dimensions during the first 6 months of the pandemic. My talk will begin by introducing the Protein Data Bank, explaining the role of the US RCSB Protein Data Bank in supporting global research and education, describing the impact of the PDB on drug discovery, and outlining RCSB PDB efforts in the time of COVID-19. I will devote the remainder of my time to presenting unpublished work on SARS-CoV-2 protein evolution in 3 dimensions, with particular focus on 2 potential antiviral drug discovery targets, the non-structural proteins 3 and 5. The Protein Data Bank was established as the first open-access digital data resource in all of biology. We are just days from beginning our 50th year of continuous operations, having been founded in 1971 with just 7 X-ray structures of proteins. Some of the structures inspiring the launch of the PDB are shown in the right panel. Since inception, the archive has grown more than 24,000-fold to become the single global resource for experimental structures of biological macromolecules. It now holds more than 170,000 structures of proteins, nucleic acids, viruses, and macromolecular machines. Recognizing the global importance of the PDB, it has been managed since 2003 by the Worldwide Protein Data Bank Partnership, encompassing the US, Asia, and Europe. The US Protein Data Bank, known as RCSB PDB, was a founding member of the Worldwide Protein Data Bank Partnership. My teams at Rutgers in New Jersey, UCSD, and UCSF in California support more than 40,000 depositors, structural biologists, worldwide. We deliver PDB data and related services to more than 4 million unique users around the world through our rcsb.org research-focused website. Our education outreach website, pdb101.rcsb.org, served more than 600,000 educators and students last year. We are jointly funded by the National Science Foundation, the US Department of Energy, and various NIH institutes. The graphic on the right celebrates reaching our 150,000 structure milestone recently. The RCSB PDB supports global research and education in fundamental biology, biomedicine, and energy sciences. We integrate 3D structured data from all areas of science with information from more than 40 external data resources to deliver knowledge through our websites to biologists, educators and students, drug hunters, and biopharmaceutical chemistry and biotechnology companies. As PDB archive keeper within the Worldwide Protein Data Bank Partnership, the RCSB has a special responsibility for safeguarding the archive, which has an estimated cost of replacement of about $17 billion US. My colleagues and I recently published 2 papers analyzing in quantitative terms the impact of the Protein Data Bank on new drug approvals by the US Food and Drug Administration. First, in a paper published last year in Structure, we showed that discovery and development of nearly 90% of the 210 new drugs approved by US FDA in 2010 through 2016 was facilitated by open access to PDB structured data. The impact of these structures ranged from target identification and validation to structure-guided drug discovery, as exemplified by BRAF kinase and its inhibitor vemurafenib, shown on the lower right. This small molecule kinase inhibitor is used to treat late-stage melanomas. In a second paper published earlier this year in Drug Discovery Today, we examined the impact of PDB data on new anti-cancer drugs approved by US FDA in 2010 through 2018. Discovery and development of approximately 90% of the 79 new anti-neoplastic drugs approved by US FDA in this time period was facilitated by open access to PDB data. We also showed that more than 70% of the 54 new small molecule anti-cancer drugs were products of structure-guided drug discovery by drug hunting teams in industry. In most of these cases, the biopharmaceutical company benefited considerably from open access to structures of the target protein previously contributed by academic structural biologists. The Protein Data Bank is widely recognized as being comprehensive, authoritative, and of high scientific quality. It has been certified by the Core Trust Seal as a trustworthy data repository and is regarded as a gold standard exemplar open access archive for biological data as a public good. As you are all too aware, the COVID-19 pandemic began in late 2019 with cases of an unexplained flu-like illness identified in Wuhan in Hubei province. By January the 10th, 2020, we knew that the causative agent was a novel coronavirus dubbed SARS-CoV-2, and the viral genome sequence was made public. X-ray and 3D EM structures of viral proteins have been pouring into the PDB since late January. The first of these was the structure of the NSP5 main protease, PDB-ID 6' uniform 7 using the NATO alphabet. More than 400 SARS-CoV-2 protein structures freely available from the PDB, and the portrait on the right painted by our molecular artist, Professor David Goodsell, allow us to visualize the so-called unseen enemy. These SARS-CoV-2 structures are being used by researchers around the world to understand the biology of the virus and discover new drugs and vaccines and characterize neutralizing antibodies. The RCSB PDB has been hard at work responding to the pandemic since the first SARS-CoV-2 protein structures were deposited. We have prioritized deposition, validation, and biocuration of COVID-19 related proteins and have strongly encouraged immediate pre-publication release of these structures. I am pleased to report that nearly all of our depositors agreed to immediate release in the interests of global public health. We have created a COVID-19 coronavirus resources page on our rcsb.org website, which is updated weekly with each release cycle for new structures. We have also developed education and outreach resources, such as the hand-washing animated video depicted in the lower right of the slide, and RCSB PDB Molecule of the Month articles featuring the NSP5 main protease, the RNA-dependent RNA polymerase, and the spike protein. The SARS-CoV-2 genome and proteome are depicted schematically in this image provided by the Swiss Institute for Bioinformatics. The viral genome is a single-stranded, positive-sense, 5' capped, polyadenylated messenger RNA ready for translation by the host cell machinery. Most of the SARS-CoV-2 non-structural proteins are expressed within two polyproteins designated 1A and 1AB. They are excised by two viral proteases, both non-structural proteins themselves and part of the polyproteins. The NSP3 papain-like or PL proteinase cuts at 3 sites denoted with dark blue arrows on the lower left. The NSP5 main protease cuts at 10 sites denoted with light blue inverted triangles. My Rutgers Institute for Quantitative Biomedicine colleague, Professor Sagar Kare, and I decided earlier this summer to use the wealth of PDB data to analyze the evolution of SARS-CoV-2 proteins in 3 dimensions during the first 6 months of the pandemic. The project team included members of the RCSB Protein Data Bank and the Kare Laboratory, and 10 summer interns, 9 undergraduates, and an extraordinarily bright high school student denoted with an asterisk. This team worked virtually through most of the summer to generate the data that I'm about to present. We downloaded 49,000 viral genome sequences from GISAID in late June and analyzed 29 viral proteins from mutations, built a 3D structural model of every unique sequence variant of each viral protein, and used Rosetta to estimate the change in the free energy of stabilization of each mutant protein. Our findings are summarized in the next 2 slides. For each study protein, each viral protein, we were able to analyze up to nearly 49,000 sequences. About 90 to 95% of the time, the viral protein sequences were found to be identical to the original Wuhan isolate. When we did detect mutations, we only saw point mutations, no insertions or deletions. Approximately 90% of these unique sequence variants, or USVs, were single point mutants. Double, triple, etc. mutants were very rare. On average, about 2/3 of the observed mutations were non-conservative. Structural models were calculated for every unique sequence variant that differed from the original Wuhan isolate. They will be made publicly available when we submit the manuscript and post it on bioRxiv later this month. About 40% of the observed mutations map to the protein surfaces. Only 7% of observed mutations on average map to the hydrophobic cores. More than 50% on average of the observed mutations map to the boundary layer between the hydrophobic cores and the protein surfaces. Rosetta energy calculations revealed that about 2/3 of the observed mutations are moderately destabilizing, which is what one would accept. SARS-CoV-2 proteins are thought to have undergone decades of low-throughput evolution in non-human hosts. About 1/4 of the observed mutations are moderately stabilizing. Finally, about 5% of observed mutations gave unreliable stabilization energies, reflecting challenges with the energy calculations themselves. I will now present 2 informative case studies of NSP3 and NSP5. First, the NSP3 papain-like proteinase. The PDB currently contains 18 structures of the PL proteinase, including both apo and co-crystal structures with a variety of inhibitors. This papain-like enzyme is part of the large multi-domain protein NSP3, which is expressed as part of polyproteins 1A and 1AB. It uses a cis-his-asp catalytic triad to cleave viral polyproteins at 3 sites, releasing non-structural proteins 1, 2, and 3. The enzyme is monomeric with a single active site and represents a potential drug discovery target. We analyzed a total of 37,181 viral proteomes with complete sequences for the PL proteinase. Approximately 97% of these sequences were unchanged from the original Wuhan isolate. 225 unique sequence variants were identified, including 215 single mutants and 10 double mutants. 30% of the observed mutations were conservative, with the remaining 70% being non-conservative. We built 225 structural models, which revealed approximately 10% of the mutations mapping within the hydrophobic core of the protein, and 50% of the mutations mapping to the surface of the protein, with nearly 40% mapping to the boundary layer in between. Most of the observed mutations were moderately destabilizing. No mutations were observed within the enzyme active site. Moving on to the NSP5 main protease case study. The PDB currently contains 188 structures of the main protease, including both apo and co-crystal structures with inhibitors and smaller chemical fragments. This chymotrypsin-like enzyme is expressed as part of the viral polyproteins 1A and 1AB. It uses a cis-his catalytic dyad to cleave the longer polyprotein at 10 sites, releasing NSP4 itself and non-structural proteins 6, 7, 8, 9, 10, 12, 13, 14, 15, and 16. The enzyme is a symmetric homodimer with 2 active sites. Like NSP3-PL proteinase, it represents a potential drug discovery target. We analyzed a total of 46,797 viral proteomes that had complete sequences for the main protease. Approximately 94% of these sequences were unchanged from the original Wuhan isolate. 217 unique sequence variants were identified, including 189 single mutants, 26 double mutants, and 2 triple mutants. Approximately 40% of the observed mutations were conservative, with the remaining 60% being non-conservative. We built 217 structural models, which revealed 13% of the mutations mapping within the hydrophobic core of the protein, and 43% of the mutations mapping to the surface of the protein, with nearly 44% in the boundary layer between. Again, most of the observed mutations were moderately destabilizing. 2 mutations were observed in the dimer interface, neither of which appeared likely to destabilize the quaternary structure of the enzyme. A single point mutant changing the histidine of the cis-his catalytic dyad was observed in a single viral isolate. Efforts are underway at Rutgers to determine whether or not this change has any effect on catalysis. The evolutionary data I presented here today for these 2 SARS-CoV-2 protease case studies revealed that viral protein mutations during the first 6 months of the COVID-19 pandemic are rare. The same was true for the remaining 27 viral proteins analyzed by the project team, reflecting the fact that all known coronavirus genomes encode a proofreading exonuclease known as NSP14. Coronaviruses depend critically on the activity of NSP14 because they have the longest genomes of any known RNA virus. Our findings also document that both NSP3-PL proteinase and NSP5 main protease active sites represent attractive, that is to say, highly conserved targets for structure-guided drug discovery. Both active sites are narrow grooves capable of recognizing and binding tightly to linear polypeptide segments that undergo cleavage after translation of the polyproteins by the host cell machinery. Inhibition of either protease would block viral replication and may be useful for treating acutely infected individuals. My final slide provides a sobering postscript. NSP5 amino acid sequences are highly conserved across all known coronaviruses. For example, the SARS-CoV-2 main protease is 95% identical to the SARS-CoV main protease in amino acid sequence. NSP5 3D structures are also highly conserved across all known coronaviruses. As you can see in the right panel, the main proteases of SARS-CoV-2 and SARS-CoV are extremely similar in 3D structure. The calculated alpha carbon root mean square deviation for this overlay was 0.8 angstroms. The active sites of the 2 enzymes are even more similar. Non-hydrogen atom RMSDs are less than 0.5 angstrom units between the two active sites. A safe and effective drug targeting SARS-CoV NSP5 would have worked for SARS-CoV-2. Investment of a few $100 million in a SARS-CoV main protease inhibitor would almost certainly have saved more than 1 million lives and prevented tens of trillions of US dollars in economic losses worldwide. Food for thought. Let me now close by acknowledging the contributions made by the enormous team of talented professionals that is the engine of the RCSB protein data bank. You can see most of them smiling here on the right. On their behalf, I thank our funders at the National Science Foundation, the National Institutes of Health, and the US Department of Energy. I also thank our host institutions, Rutgers University, and the University of California, San Diego, and the University of California, San Francisco. The RCSB PDB is a member of the Worldwide Protein Data Bank Partnership. Finally, I thank you for listening and the organizers for the honor of representing the RCSB Protein Data Bank at this joint Academia Sinica CODATA conference. I wish good health to you all.
Thank you, Stephen. I understand that you have returned online with us. So you can. Thank you for staying awake with us. I understand you are 2 o'clock past midnight. So would you entertain some questions from the audience?
Of course, Lao Wang, thank you.
Okay. 他叫我老王。Any question from the audience? Related to your last sobering issue, you know, about 10 years, more than 10 years ago, I worked on the 3CL inhibitors, and it was a good inhibitor, but then SARS went away, disappeared sort of all of a sudden. So we stopped the study, and it was kind of unfortunate. If we push it all the way through, we would have drugs probably available now. So with that question you raised, do you think the government or whoever may wake up to support this kind of research continuously?
I hope so, Andy. I wrote a piece that was published in Nature, a worldview, arguing that the free market has failed the world in terms of emergency drug discovery, and that we needed a new approach that would involve governments, industry, and non-governmental organizations to make investments in rational approaches to pathogens such as the coronaviruses. I think that given how closely related the NSP5s are among all the coronaviruses, it should be possible to make a handful, perhaps, of inhibitors that have good drug-like properties and good efficacy against the different clades of NSP5, if you will. And those could be tested in animals and then ultimately tested in normal volunteers. And when the next coronavirus crosses the species barrier, does so, we would be in a position, conceivably, to actually be able to ramp up production of an inhibitor which would serve as an effective drug. The cost of doing this would literally be a few $100 million. And juxtaposed to the massive losses in terms of life and the global recession that we're now in the midst of, that seems like a very good investment. I think it's probably fair to say that after SARS went away, that the decision to abandon work on anti-SARS agents might have been rational. But after MERS, I would argue that it wasn't rational. Once MERS had happened, particularly given how deadly MERS was, I think that a concerted effort should have been made at that time to look for a general approach to anti-coronaviral agents. And I regret that I didn't publish my Nature Worldview in 2013. I only published it in 2020.
Thank you. Well, in view of the time, unfortunately, we have to stop here. But Stephen, I still look forward to you coming to Taiwan, visit in person. So hopefully,
thank you. I look forward to the same.
Yeah. Okay. Thank you very much.
All right.
Goodbye.
Thank you. 再见.
Okay. Let's move on to the next speaker, Hui-Ling. She is currently a associate professor, School of Public Health at the University of Hong Kong, and she has been doing well. Let's see, the epidemiology studies of various kind of virus, but today I believe she's going to talk about the COVID-19 virus somewhat. So let's, oh, she's also going to be online, right? Yeah, okay. So let's start the video. Thank you.
Good afternoon. I'd like to thank the meeting organizers for the opportunity to participate in the International Symposium on COVID-19. Today, I'd like to share our research on using the golden hamster to study pathogenesis and transmission of SARS coronavirus 2. Previous studies on SARS coronavirus have shown the importance of spike ACE2 injection and age and innate response in determining the disease severity in animal models. In inbred mice, we know that there is replication, but without apparent histopathological changes. Although in the aged inbred mice, we can see more weight loss and more severe diseases. Interferon response have been shown to be critical for viral clearance because the step 1 knockout mice were shown to have prolonged viral shedding and extrapulmonary spread. Overexpression of human S2 in the mouse model may support better viral replication and more prominent histopathological changes. However, this is also accompanied with overexpression of the S2 in the mouse brain, and lethality were often due to extensive neuronal infection. Hamsters have been shown previously to support robust viral replication in the lungs, which leads to pronounced histopathology changes. But there was no apparent weight loss or clinical signs. Multiple non-human primates were also shown to support viral replication and pneumonitis. And in this animal model, there is no apparent to mild diseases, but there is no severe clinical signs or lethality. In addition to that, ferrets and cats have been also shown to support replication. Earlier study this year done by Zhou and al. Have demonstrated the importance of human S2 in supporting viral entry. So this is the HeLa cells overexpressing either human S2 or murine S2. You can see there is a different response for viral infection. We analyzed the S2 sequence of 4 different animals, the human, the macaque, the hamster, and the mouse. Because there are 20 residues known on the S2 protein to interact with the spike protein. The macaque is showing identical sequence to the human S2 at these 20 residues, whereas there are 2 mismatch between hamster and human, and there are 7 mismatch between mouse and human. So this alignment suggests that the hamster may be a better model than mouse in supporting replication of SARS coronavirus 2. We started by inoculating male 4 to 5 weeks old hamster intranasally with 8 times 10 to the 40 CID50 of the SARS coronavirus 2. And on day 2, day 5, and day 7, we euthanized the animals and monitor viral replication and histopathological changes. We can observe very robust replication early after inoculation with the highest infectious virus titer detected on day 2, as shown here in blue color. And you can also see that the infectious virus were rapidly cleared by day 7. However, viral RNA can be continued detected. On day 2, we can see that viral antigens were predominantly infecting bronchial epithelium cells, with about 10 to 15% of lung consolidation. On day 5 post-inoculation, we see now with increased lung consolidation, and then now the antigen-positive cells have moved on to the pneumocytes. There is also increased immune cell infiltrations at this time point. On day 7 post-inoculation, we can see further increased lung consolidation, but interestingly, there is no more antigen-positive cells being detected in the lung. On day 5, we're also detecting an increased number of CD3 positive cells in the lungs, and this is also including detection of hyperplasia of pneumocytes on day 7. We also observed that the SARS coronavirus 2 can effectively infect nasal epithelium cells. As shown here in the middle panel, you can see that the antigen is now shown in brown color with the N protein being detected. In addition to the nasal epithelium cells, we see some positive cells showing the morphology resemble the neuronal cells. To confirm if neuronal cells can really be infected, we perform additional staining. So here on the right-hand side, you will see this panel with double staining. Now the SARS coronavirus 2 N protein is shown in pink color, whereas the neuron-specific beta-3 tubulin is now stained in brown color. And many of the cells were stained positive for both markers. We also perform time-dependent observation of the nasal epithelium cells. And we can see that now this is detecting neuron-specific beta-3 tubulin in brown color. On day 0, there is a nice layer with positive cells detected by the antibody. On day 2, we can see already a loss of the neuronal cells. And then by day 7, there is a prominent nasal epithelium attenuation. And by day 14, there is a rapid repairing. Outside the respiratory tract, we detected viral antigen in the duodenum. This is accompanied by detection of viral RNA in the fecal samples. However, we were not able to detect any antigen-positive cells in tissues including brain, heart, liver, or kidney. Although we can see there was a transient detection of viral RNA in the kidney early post-inoculation. We then went on to test if the SARS coronavirus 2 can be transmitted in hamster. On day 0, we inoculated hamster with 8 times 10 to the 4 TCID50 intranasally. We then introduced a naive contact hamster to co-house with the donor from day 1 to day 14. This diagram shows you the viral shedding in the donor nasal washes, as well as the weight changes of the donor over time. You can see that the infectious virus was the highest on day 2 post-inoculation. This is shown in the bar. However, the infectious virus was not detected already on day 6 post-inoculation, suggesting that there is a very rapid viral clearance for the infectious virus. However, you can also see the dots, which represents the viral RNA detected in the nasal washes, and this continued to be detected for a very long period of time. Maximum weight loss was detected on day 6 post-inoculation, with about 10% to 15% weight loss in the donors. Transmission to the contact was also very efficient. We can detect virus in the contact animal nasal washes already on day 1 post-inoculation. Again, this is followed by a very rapid viral clearance. So by day 7 post-contact, there is no longer infectious viruses being detected. Weight changes follow a very similar pattern as the donor, with maximum weight change detected on day 6. So in this experiment, we detected transmission in 3 out of 3 pairs of the contact animals. And we can also detect post-contact serum with positive antibody response in the contact at 1 to 160, 1 to 320, and 1 to 160, neutralizing antibody titers. So here I'd like to highlight that the detection of infectious viruses was only for a very short period of time. And by day 6, we noted that the donors was no longer shedding infectious viruses. We were curious whether this could lead to differential transmission potential. We then repeated this experiment. So instead of introducing the contact animals on day 1, we now introduce the animal on day 2 to co-house with the infected inoculated donor. And these animals will continue to co-house from day 6 to day 20. And here you can see that none of the contact animals actually was shedding infectious virus in their nasal washes. And all three contact animals gained weight over the observation period. We also detected no seroconversion of those contact animals, as none of them had neutralizing antibodies after exposure. So this experiment suggests that the communicable period was short and correlated with the detection of infectious virus, but not the viral RNA from the donors. The finding is in line with WHO's updated recommendation on the criteria for releasing COVID-19 patients from isolation. Transmission by direct contact can be actually mediated by multiple different routes. So transmission by aerosol is one of them. We then continue to test if an inoculated donor is able to transmit the disease to a naive animal through air. So in this experiment, the donor was inoculated, and then the contact animals were introduced on day 1. The donor and the contact animals were housed separately in 2 containers, as you can see in the diagram here, which allows the air to pass through, but without any physical direct contact. And the aerosol contact animal was only exposed for 8 hours within this ISO cage with the donor. After 8 hours, they were all separated and housed individually in the individually ventilated cages. This diagram shows you the donor viral shedding pattern as well as the weight changes. Again, you can see that viral clearance occurs very quickly, although the viral RNA can be continually detected for a long time. Weight changes again was maximally detected on day 6 post-inoculation. We observed very effective, efficient transmission by the aerosols to the contacts, as you can see here from the viral shedding. So on day 1 post-inoculation, all 3 contact animals start to shed virus in the nasal washes. We can also detect viral RNA being shed by the contact animals in the feces, but no infectious virus was detected in the feces. So in this experiment, we detected 3 out of 3 transmission events in the aerosol contacts. Post-exposure sera, we can detect antibody at 1 to 160. To test if the animals can be protected by the antibody response that was previously acquired after natural infection, we challenged aerosol contacts on day 49 post-contact, again with the 10 to the 5 TCID50 of the SARS coronavirus. You can see that there was some RNA being detected and some very low titer of infectious virus detected on day 2 post-inoculation, but there is no continued replication in the nasal washes, whilst the changes remains in the weight remains unchanged, suggesting that the aerosol contacts who have previously developed neutralizing antibodies after infection now are being protected upon re-challenge. We then went on to test if transmission can be mediated by fomite routes. We inoculated donors on day 0 intranasally with 8 times 10 to the 4 TCID50. And this donor was housed inside a cage for 2 days to create a soiled cage. On day 2, this donor is then removed. We then housed a naive contact animals inside this soil cage for another 48 hours. Afterwards, this animal was moved out to house in another cage. To confirm if the cage was really contaminated, we sampled different surface area within this cage, and we can detect infectious viruses as well as viral RNA from these cages. With this experimental setting, we can see that there was 1 out of 3 events of transmission. And then you can see that the infected animal was able to shed virus in the nasal washes and in the fecal samples. Whereas the other 2 animals, there was no detectable viruses and continue to gain weight over time. We also re-challenged these 3 contact animals on day 48 post-contact. So just to remind you that only 1 animal had developed antibody post-exposure, whereas the other 2 did not develop any neutralizing antibody in the previous experiment. So upon re-challenge, as expected, we can see that the 2 animals previously uninfected now become infected with the virus, with robust viral replication and viral shedding and weight changes. Whereas the animal that was previously infected and successfully developed antibody response now is being protected. So a brief summary on the pathogenesis in hamsters. We noted that viral replication was very efficient in the respiratory epithelium cells, including the nasal mucosa, bronchial epithelium cells, and the pneumocytes. Antigen was also detected in the duodenum, but no infectious virus was detected, although RNA can be continuously detected for a prolonged period of time. The ability of the SARS coronavirus to infect the olfactory sensory neurons may explain the transient anosmia reported in COVID-19 patients. Higher viral loads were generally detected early post-inoculation, followed by rapid clearance of the infectious virus. However, viral RNA can be continuously detected. Infection in the golden hamster resembles that was found in humans with mild COVID-19 symptoms. In regards of transmission, we know that the virus can transmit efficiently from the inoculated donors to naive contacts by direct contact. But the communicable period was short. We show that this is less than 6 days and was associated with the detection of infectious virus, but not viral RNA. Transmission by aerosols was highly efficient, with transmission detected in 3 out of 3 contacts. The relative significance of large droplets and fine aerosols in mediating transmission of SARS coronavirus remain to be studied. On the other hand, transmission by fomites was less efficient, with only 1 out of 3 transmission events observed. Infected animals were actually being protected upon a re-challenge experiments. So more recently, we've been in collaboration with Dr. Ali Elabedi from Washington University, trying to understand the effect of neutralizing antibodies in protecting against SARS coronavirus infection. So Dr. Alibelli have generated murine monoclonal antibodies by immunizing the mice with RBD protein, and then followed by 2 boosts of the spike protein. He then sorted out the plasmablast, particularly the RBD binding plasmablast, for cloning and generating monoclonal antibodies. So total there were 26 murine human chimeric IgG1 mobs generated, and this would bind to RBD. Among them, there was a particularly one monoclonal antibody highly potent. This is the 2B04 clone with an IC50 at 1.46 nanogram per mil. So we received this antibody from Ali, and then we use it to test the prophylactic and therapeutic efficacy against SARS coronavirus 2 infections in hamsters. We give hamster those antibodies by IP injection, so 1 milligram per hamster, either 1 day before inoculation or at 16 hours post-inoculation. On day 2, day 5, we then monitor viral loads in the nasal turbinates and in the lung. We can see that both prophylaxis or therapeutic treatment was able to reduce infectious viral load in the nasal turbinate, although it was not a neutralizing activity, complete neutralizing. And also in the lungs, we can see that either prophylactic or therapeutic was able to, again, reduce infectious virus titers in the lung, and also viral RNA. So on day 2, day 4, day 6, and day 8, we also collected nasal washes of those animals. Among the isotype control group, as you can see here, replication of the virus was very robust, with the highest titer detected on day 2. And we can see the maximum weight loss at day 7 post-inoculation. Prophylactic treatment was quite effective, and you can see now the day 2 viral load was significantly reduced. Unfortunately, there was no complete neutralization of this replication, so there was still residual virus replicating. However, from the weight changes, you can see that the animals are actually protected. For therapeutic treatment, on day 2, you can see that the viral load on day 2 was higher compared to prophylactic treatment. However, it is still lower compared to the isotype control. Also from the weight differences, we can see that it is still protected compared to the isotype control group. So we show from this experiment that the 2B04 is a potent antibody, neutralizing antibody against SARS coronavirus 2 with the IC50 at 1.46 nanogram per mil. We show that the prophylactic treatment was significantly reduced viral loads in the nasal turbinates and the lungs after inoculation, and also prevented weight loss. Therapeutic treatment moderately reduced viral loads in the nasal turbinate and the lungs, and may also reduce weight loss. Finally, I'd like to thank the funding provided by the Hong Kong government, as well as from the SEARS network, from NIAID, NIH, and also the excellent teamwork and collaboration from the important collaborators as listed here. So in particular, I'd like to highlight the Professor Zhang Nichols, who is the pathologist helping us to do the immunostaining, Professor Malik Paris and Leo Poon for support and comments, and also Dr. Ali Badi for providing the monoclonal antibodies. Thank you for your attention.
Thank you for your nice talk. In view of the time, we have one question from the audience. Yes, Jason?
Yes, great, wonderful work. Thank you very much for sharing the results of the animal studies. I was wondering, Currently in the United States, when patients are diagnosed with COVID, we ask them to stay home. Do you think we should wait them? Has there been any any human studies on weighing patients during the self-isolation period to see how they're doing? Do you have any data from Hong Kong that weighs people?
Thank you for the question. Yeah, so I don't have any data on patient weight changes. So yeah, I think it's a great suggestion. But I think for us, for animals, it's of course very difficult to ask them how do they feel. So weighing them is probably the only way that we can tell whether they are really sick or not. For most of the cases. For human, I think it's easier to assess whether they are getting better.
Yeah, currently in the United States, we do not have that capacity to call everybody. So maybe we should ask them to wait. But the second question I have for you is that how did you figure out for the aerosol study, it'll be 1.8 centimeters between the animals?
Yeah, that's also a great question. We are actually restricted by the size of the ISO cages we have, and that's the reason they were only separated by about 1.8 centimeters. This is of course not the ideal situation. We should probably have tested them with different distances. So in the past, our laboratory have experiences working with influenza. We actually have tried to understand the particle size at different size range that mediate transmission in the ferret model. So we are going to perform similar studies with the hamster in the coming month.
Okay, thank you. I know there are other questions, but we are running out of time. So if you have a question, you are welcome to submit the question and we can send it to her via internet. Okay, thank you. Thank you, Professor Yan. Okay, next speaker is Dr. Ching-Yung. He is an expert in AI applications. He worked previously also at IBM, T.J. Watson Research Center, and so on. He developed a very sophisticated algorithm to track the terrorists around the world. And now he used this to track the biological terrorist, the COVID-19. So let's hear what he can tell us. Please go ahead.
Good afternoon, everyone. My name is Ching-Yung. I'm from Graphen and also Columbia University. So it's my pleasure to present our work on dive monitoring of a SARS-CoV-2 evolution on this afternoon. First, you can show this video that was recorded from our visualizer. So it actually shows this kind of the early stages of the SARS-CoV-2 mutations. And you can see how it grows through time, like originally from a small one, and then gradually bigger and bigger and bigger. I think you all know the story. And here just a visualization try to visualize what happened through time. And you can see it just keep growing bigger and bigger. And we can see the detail one by one. So for example, here here is a visualization for the the early stages. So it's in the December timeframe. So at this time, you can see that originally there is one variation here, and so the green dot actually present one type of the mutation, and then the blue dot, the red dot means that on the same viruses that was detected with the same mutation. And you can see like the early stages that there's one type in the middle. It was detected for 1, 2, 3, 4, 5, 6 times. And then you can see it's mutated to another one, several of them, the green ones. And then each one of them, you can see some samples on them. And you can see that by January 31st, the virus is already evolved to like 2 major groups. You can see the one in the middle, So the one in the left-hand side, that is the biggest one. And you can see on the right-hand side, gradually is kind of there is a circle there, and then you can see several of the changes there. So these are the one happened in January. And as the time goes by, you can see that in February, there are more and more clusters coming up. So you can see they are very distinct variations. So each one of them, you actually can see their exact variations and also the samples on that. So that time we actually created the system and put it online to the everybody can access to see what's going on for this COVID-19 virus. And these are example. These are exactly what happened in March. So by March, you can see that there were 2472 sequence viruses. So these viruses sequences they were contributed by the worldwide labs. And you can see that among these 2400 viruses, they actually belong to 1237 different type of variations. So that is about like you actually can see the number of the sequences and the number of the cells usually is in the range of 2 versus 1. So you can see usually how the viruses their changes and then how they are different from this type of ratio. But please also note that a lot of this set actually it probably only happen in one person. It may not be propagated. So if it's propagated, usually it means that those variations are kind of stable. Okay. So by the end of May, you can see there are already 34,000 sequences of viruses, and the variations kind of kept the ratio about 2 to 1 ratio on that. And when we analyze this, when we created a slice by the data from September 28th, so there was already 106,000 sequence of viruses. So this has been sequenced with their full genome has been sequenced there. And we actually created a work, try to analyze then combine several different kind of methodologies. So these are key technology are including the large-scale grid database. So large-scale grid database is the one try to combine all the information together. So as you can see that this is probably the first case in the world that you have the one virus that is a whole mutation has been sequenced by the big number. So remember last time it was I think SARS there was only about like in the tens to low hundreds of the viruses were sequenced at the time. And at this time you can see this scale is actually kind of big. So if you really want to handle this type of big relationship, you need this type of large scale grid database to do the work to put them together. Then you are able to kind of analyze them one by one. And we also create a real-time system that are able to monitor this kind of situation. When the data are coming in, then we are able to analyze that automatically. So then therefore you can keep up-to-date situation for the live monitoring. And the third key technology we apply is graph spectrum clustering. So this has been the technology applied to make finding out the key clusters and how they cluster each other. So it helps you with this technology, you can easily see the families of the viruses, and also we can use that for visualization, because that when the size is big, it's not possible to visualize everything. So consider like a 106,000 nodes in a screen. It's not possible to visualize that. So you need all kinds of hierarchical clustering technologies, and then to be able to find out the key foundations on those kinds of distributions, and then therefore it becomes possible to visualize that, to see what's going on there. So that is actually also another important thing that we apply to help make this happen. So Let me introduce them one by one on these key technologies. So the major technology has been based on our AI platform. So this our AI platform actually include a lot of the different kind of the methodologies, and we actually built that. In terms of this AI platform, we built it for the finance applications, for medical applications, and for the security applications. So this type of applications, we actually deployed that in New York, in Shanghai, Hong Kong, Taipei, in these areas. And the key part of this platform is that it tries to simulate the full plan function. So include a large scale database to be able to handle the memory part, to find the contextual memories, to find out contextual information, how one is correlated with the other one. And then including the sensor information, try to sense the all kinds of the signals. And then you do the recognition to understand what's going on. And then it try to do the perception comprehension about the causalities on that. So this is a kind of the graph computing based platform. It try to simulate the human brain, since our human brain is actually a brain network of about 100 billion nodes and 700 trillion edges. And another key part of that is that we built that to be able to handle large scale, to be stable, to handle all kinds of applications. So it is actually something like this one. So the live data coming in, and it was stored in our graph database. And then graph database will have the graph analytics, try to process them. And so for example, do the spectrum clustering, or try to find out what are the key bridges and what are the key nodes on the whole graph. So therefore, it's a way to find out something like a centrality. So this kind of centrality analysis is an important way to find out how the it can be used for information flow, like we previously applied it on social media, or it can be used for this kind of situation, the virus propagations. And then we can use a machine learning model and then try to find out this kind of causal relationship, like how one mutated to another one, and to find out this kind of parent-children situations. And then therefore we can apply that in our the pipeline to build that and put it on the internet. So this type of graph analysis has been a key for us to be able to propagate it, to find out the knowledges and intelligence. So previously we actually can use this type of technology to highly improve the way that the behavior prediction, and it actually can be like 10 times faster compared to the traditional machine learning technologies. And so here we applied it to analyze the genome of this COVID-19 virus. So here that you can see an example, I believe you probably also hear that from several previous talks on the genome of this SARS-CoV-2 virus. So there are about 30,000, near 30,000 RNA letters, and then they are actually, it can be divided into around 26 proteins. And you can see that including ORF1A, actually can be divided into around 16 NSPs, and then the key structure proteins, the spike, E, M, and N, and then also other auxiliary proteins in between these structure proteins. So here is a distribution of the the mutated strands percentage. So you can see that how each proteins, how much they are changed through the entire 106,000 sequence of viruses. As you can see that like the S protein, the mutations on S protein in this 106,000 viruses has been near 90%. So like NSP3, you can see is also near 90%. And then you can see also the ones that are very few variations, like the E protein is among the lowest one. So for that one, that is actually is usually is like in the middle. So it's quite it's an important one that it does not mutate much, and the S usually will mutate a lot because that is one has a lot of the stress to be adaptive to the animals it try to hinge to, right? So that's why that S protein actually is usually stress on evolution. But the E is actually not. E is more stable. So it's fine. They actually have a different purpose there. And you also can see some of them are also small. The variations are small and some are very large. So here you can see how the distributions are in terms of the mutations. And we can also check that the types of the missense mutations in each protein. So you can see that these are the ones that we try to check how many different type of mutations in each protein. So as you can see that the S protein actually also have about near 600 different type of the mutations on that. So overall, I think S protein only have about 1,200, 1,300, the type of the number of the different locations. And you can see with the 593, I think it's 593. With that number, it's almost like half of the locations in the S protein can possibly mutated. So it's actually a lot already. Okay, and here you also can see that we try to find it out because, as I said at the very beginning, a lot of the mutations it probably only happen once, only in one person, because it kind of happen in a person's body when you take it out, try to do the sequencing, it probably has some mutations itself. So, but the mutation is not stable, so you will not like propagate out, but it will be in the person. So that's why a lot of them are a lot of the mutated ones are actually quite. The numbers have been quite small. So in this case, we try to see if we take care of the bigger one, the more popular ones, then how many different types. So we check the number like these 10 mutated strands, and we can see that even in the sense that S protein actually has 354 type of the popular mutated strands. So here popular would mean that these are 10. And we can also see there are 28 different types in the E mutations. So here are actually the importance are, there are two types of importance. So like S actually is quite useful for the machine, and the E is actually quite useful in the detection. So that's why that you can see the number of the types that they keep growing, then you need to consider this when you develop your machine and also the ones who want to do the detection. So here is the S protein structure, and then we just show an example that as you can see, on top of it is actually the RBD area that they try to link to the ACE of human being, the ACE2, and here we just show some kind of colored areas. Those are actually probably some of the newer. It may be come from recombination or from other reasons, or the newer things that are not previously shown on other PCBs. And the RBD ones are actually more better studied. And then there are still some kind of portions that are not well studied yet. But here is kind of roughly where they are on this new changes on this virus. The most popular mutation has been the D614G. D614G is kind of famous. I think everybody right now almost know. Is actually 87.23% of the worldwide viruses actually belongs to this one. So it actually coming from originally it happened somewhere in China, but the mutation probably didn't propagate along until a person fly to Germany, and then it start to propagate in Germany, and then eventually it goes to the whole Europe, and then the US, the East US, and then other places in the world. And so it's why that eventually this number actually grows up and up through time. So the one D614G is famous. So I think most of people know that they need to take care of that when they do the machine development, especially right now, they're actually the major ones right now in the world. However, there are a lot of the others actually also probably also need to be considered. So as in this figure I show in the log scale, there are also the ones in several other areas in the S protein. And so the ones that we may need to consider is this for the ones in the RBD area, because in the RBD area that you can see, for example, the S477N is the one that I think the next popular, or the next popular mutations after the D614G, and then I don't see any or much of the the discussion on this one. So this one actually it originally started from in England in April, and then it became quite popular in Australia. I'm seeing that in Australia that a lot of the viruses actually right now, including this one. So in fact, this one actually already have like I think 3,000, 4,000 in the range already. So it's actually I also saw it the kind of the become yes a lot of the increase in late August or early September, that time frame. So you can see that it actually becomes popular in some area of the world. So in other words, if you consider the vaccine, probably, for example, in Australia, this may be something you need to specially consider. And you can also see other changes, right? So here I try to highlight like the one that in red. The one in red, these are the 2 points among 5 or 6 exact points that are the SARS-CoV-2 bind to the human HEE2. So these are the exact points that bind. And those exact points that they bind, that we also see 2 of them as a kind of mutations. And one way to check whether they may be have some impact is try to finding out the distance between the similarity of these changes. And then with them, here's a chart to compare different amino acids changes, the distance changes. And then we can see that like I actually added the distance number here. You can see like N501Y, for example, when it was changed from N to Y, and it's changed in the binding point, you probably can kind of well predict this one probably has a real changes on this virus binding behavior. And then also you can also see like the other 486, the 22 is actually a smaller number, so it's probably may not change that much. So I think it depends. So these are some examples about how you can consider and whether some of the new ones actually are emerging. Okay. So we are keep special monitoring on some of the hot areas that are probably have some kind of importance on the drug and machine development, for example, like this. And another important one that we have been showing is the one that how do we do the clustering? How do we find out the family? So the major technology we use is graph spectral analysis, and we develop a new algorithm to be able to find out what will be the optimal choices on the number of clusters based on the graph, and then what will they be? So we can automatically find it out. For example, by February 29, it was a kind of the 4 major clusters, and then we can see their colors there. So these are all done automatically by our algorithm. And then my March 31st, the optimal choice of the value of clusters is 7, as you can see on the color. So this color kind of show you, kind of visualize you to help you kind of fairly easy to be convinced that these are the number of the clusters there. And then April 30th, you can see the one on the hand side. And 34th, you can see the major numbers, k equals to 10. Okay. So based on those clustering, we actually can try to find out there were kind of the 8 major families there from those clusters. We kind of combine 3 small ones into a major, into a bigger one, because those are, some of them are actually too small. So so that's why at the end we chose there are about actually 8 major families there. And these 8 major families, you can see that they are actually the A is kind of the origin. So you can see there are actually A1 and A2. And then B is the one mainly propagated outside China. And then when the D614G first appear in Europe, then it kind of begin in Europe there. And then later on, it kind of keep evolving to, here we call it G and H, that evolve from the C cluster D614G. And then the G and the H, this actually has 2 distinct variations. The G actually has a kind of 3 consecutive changes in the N protein. And then H is the one has a ORF3A protein variation that is potentially has some special meaning of that. I will introduce that. And then the D, E, and F, those are the one that are kind of propagated to other parts. I think especially the E and the F are the one propagated to the, it was propagated from the B, and then it went to the went to other places like the Spain, Australia, South Korea, China, and etc. So it's actually like this. So A becomes E, so A becomes E and the F, A2 becomes E and the F, and A1 becomes B, C, D, C becomes the G and the H. Okay. So the first set is A and the B. So A1 and the A2 are 2 origins there. And we actually can find it out from the data to be able to kind of clearly finding out, like by the first sequence number, December 24, 2019, there were actually already kind of 3 to 5 variants in various genomes. So in other words, because later we found out this kind of the SARS-CoV-2 as a stable mutated point per week. Therefore, you actually can kind of try to go back at the time to kind of to realize that the earliest case start propagating in mankind is probably around later May, late November or early December. This kind of time frame in 2019. Okay. And here is just a reference that interesting points that the virus actually the first appear, a lot of them is in this Huanan seafood market. The Huanan seafood market is actually quite close to the Hankou, the train station, and this area is actually far away from the Wuhan the virus institute. Actually, it's a kind of in quite other side of the Wuhan, so it's actually there are some distance on that. And but it's also another interesting thing. There are also some kind of a disease center actually around the Hankou area. So so those are the kind of the places that are first reported with this virus. And we actually created a website that were able to show you the kind of the life changes, and you can actually check the different type of the viruses happened in the world or in any continent, or in any country, region, or in the city level. You can even check in the city level. So if you're interested, please welcome to visit our website. And this actually a dynamic changes on these numbers on that. You actually can see that. And so I think you can directly go to our website to see the changes on the like different type. So you can see that every day. Okay, and I think for the sake of time, let me skip that. Here you actually can kind of help you to see. On the website, we also show you the different type of the families in different places, and you can see that in below the world map web page, and you can see the different changes through time. So, for example, the one in China, you can see that originally Wuhan is this kind of the A and the B type, and then Beijing is actually quite diverse on different type of them. In the US, you can also see like originally in the West, there was kind of the virus was it came from China, and but later on in the East US has been kind of dominated by the virus actually originally from France, and for that virus actually it's classified as H. It kind of become popular, especially like in New York area. There it was kind of about 90% of the viruses found in New York belongs to this one that originally coming from France. And that one actually become popular, more and more popular. It actually become the majority in the US right now. Okay, and now you can see that Japan is a different one, and Italy are more like the original, or the one from origin in Europe, the D614G, and the Brazil is already has a variation there. So as you can see that the different places, the popular ones actually are different. And yeah, the US are just like being getting viruses from two type of sources. And the one particular in the East US has been the one is actually has a special mutations in this ORF3A. So ORF3A is actually the protein to help to escape. So when you duplicate, you try to break the cell, and then try to go it out. So this one, key mutation here is actually on the one is kind of a trio outside. So this one probably also can, it might be some reason also help to the fast propagation that we found in the East US. It actually is kind of even stronger this capability to the D614G that I find in Europe. So overall, that we try to build this tool to help to understand the detect virus, diagnose its evolution, and check its geographical footprint, and also see how it changes. And so this was actually a white paper try to analyze the pandemic impact on global R&D on the AI medical, and they expect more and more money will be specific on the area. And they actually mentioned us together with Alibaba and Google, and also mentioned that tools from Graphen, Google, NVIDIA, Intel actually will accelerate the speed of drug discovery, development, testing. So that's how we hope that our tool can help to contribute. We can contribute in this area. And we also have ongoing work is try to help to do the human whole genome sequencing service. So this part of the work can potentially also help each individual can have a kind of understanding whether you are also have some kind of high risk in some of your gene changes in some places. For example, there were studies that in chromosome 22 that here is where your ACE2 gene is, and previous study from the data in Italy actually showed that for those that has more serious cases, that they tend to have less variations in the ACE2 gene. And another interesting study was that the chromosome 3, there are also some kind of Neanderthal genes that if you inherited that, then you actually may have a higher risk on your symptoms for the COVID-19. So these are actually some of the areas we are actually also studying, try to also provide this kind of service. So I think that this kind of conclude my talk. And yeah, so welcome to contact me if you have any questions, suggestions. I think there are some areas we actually can collaborate to work together to help on this kind of monitor what's going on, and we will be happy to contribute to the drug machine developments for this. Okay, so thank you for listening. Thank you.
Thank you, Dr. Lin. Dr. Lin is online to answer questions in case the audience has a question in view of the time. Oh, yeah.
Very nice talk. And I'm wondering whether you tried a similar system to analyze another RNA virus, such as influenza virus, because as we know, also mentioned by Stephen, the coronavirus is relatively stable.
Did you get the question, Dr. Lin?
So, thank you for your question. So, no, we have not analyzed other viruses yet. So, probably we can start like analyzing influenza, like for example, like what you mentioned. Yeah, since right now it's about the season, so maybe we can start analyzing that.
Any other questions from the audience? I have one question from your analysis. You pointed out that D614 to G mutation is very prevalent. Is there a particular reason why this amino acid is a hotspot from your analysis? Yes.
Yeah. So there was some of the study mentioned that because D614G, it may help the separation between S1 and S2. So once that the virus is entering the cell, so it is some suggestions suggest that this one can help the separation, so it can make the attachment much more efficient. And yeah, so there are several studies on that. So those studies actually suggest that it may be even like 10 times more possible to infect people.
Okay, thank you. Well, in view of the time, we'll have to stop here. And let's thank Dr. Lin again. Thank you.
Okay, thank you, everyone.